This is CSaP's Science and Policy Podcast from the University of Cambridge, where we're bringing you the latest evidence and expertise to improve public policymaking. So welcome, I'm Rob Doubleday from the Centre for Science and Policy, and this is the first episode in our new series, which is looking at the science and policy of genetic technologies. I think it's true to say that our societies are increasingly dependent on technologies, and not just a set of individual technologies, but the complex interaction among technologies. And that really puts expertise really at the heart of how we lead our lives, expertise that's needed to develop the technology, to run them, and also to regulate them. This raises the question about democracy and public deliberation. How is it that, why does society get a voice in thinking about the technologies we live with and the direction of their development? And that's what I'd like this series to open up, a discussion about where technologies are going, what options there are, and how they're regulated. We're looking particularly at genetic technologies here, and we're trying to bring interdisciplinary perspectives to enrich our understanding of what is going on, what it means, and what possible futures we can expect. In this first episode, I'm delighted to introduce Jonny Hazell, who's a senior policy advisor from the Royal Society who leads their genetic technologies work. Helen, a senior lecturer in the history of science from the History and Philosophy of Science Department in Cambridge, who leads a very interesting Wellcome Trust-funded project looking at historical perspectives on crop diversity and food security. And Giles Oldroyd, who's a plant scientist and director of the new Crop Science Centre at Cambridge, which opened just last year in 2020. And Giles's research particularly focuses on the interaction between plants and bacteria and fungi and how to understand those interactions in the lab and also in the field. So three great people to open the discussion with. Turning first to you, Jonny, why has the question of genetic technologies in agriculture returned again to governments at the moment?
This is partly technology driven. Since the noughties and the early 2010s, we saw this emergence of a new class of technologies for changing DNA, which are referred to as genome editing. These technologies raised new questions about the regulatory framework for technologies used in agriculture. Previous generation of technologies for genetic modification all involved moving genes between species. And so there was a regulatory framework that developed on the principle of moving genes between species. And if you use a technology that changed DNA within a species, that wasn't treated as genetic modification. But if you moved a gene between species, that was. And genome editing blurred that boundary because you could use it for both purposes. So partly the question is driven by the technology. It's also driven by societal needs. And the scientists working on these technologies are using them to address questions that government is interested in. How can we produce more food with less environmental impact and less artificial inputs? So there has been a desire on the part of scientists, on the part of agricultural companies, on the part of farmers, to be allowed to use these technologies because they see them as answering some of the problems that agriculture faces.
How can we understand the controversies around GM organisms in food and agriculture in the late 90s and early noughties? Why was it that, in your opinion, GMO became such a controversial issue in the late 90s and early 2000s? So
I think there was a coincidence of the emergence of the commercial use of GM technology. So the first commercial GM product was a tomato in the mid-90s, Flavor Saver tomato. And a sense that these technologies had been rushed from the lab to the field without much of a public debate about whether they were desirable, which coincided with a serious food systems crisis, particularly in the UK, around mad cow disease. And that had a huge impact on public trust in the governance of agricultural technology. And so whilst questions were being raised about genetic modification, there was also a sense that you couldn't necessarily trust the government and the companies involved in producing our food to look after our best interests. And so particularly at that point, genetic modification was only being used by the large agrochemical companies, so the Monsantos of this world, who already had a bad reputation the way some of their products have been used. So I think those circumstances combined to tarnish GMOs by the same brush.
And so what would you say, looking back, do you think was some of the main concerns or motivations that, if you like, mobilized public concern or controversy?
I think that there was a sense that this was another under-scrutinized, possibly risky, used for purposes that I wouldn't necessarily agree with technology.
Would you think that it's also questions of sort of power, if you like, who is going to benefit from this?
Exactly, who was using this for what purpose and who benefited? And if a lot of the purposes were about making crops that were compatible with herbicides, and I was skeptical about the use of herbicides in agriculture anyway, then I wouldn't be wild about a new technology that made it even more desirable to use herbicides.
And of course, we're kind of contracting, years of debate and contest and regulatory innovation. But could I ask you to sort of summarize what has emerged as the kind of regulatory response in the UK and in Europe to that series of issues, as you say, that arrived at a certain kind of point in history?
So the regulatory response was to try and wall off that form of genetic modification. So as we'll hear about from Helen, science and technology has long been involved in the generation of new crops. There are many ways that we have sought to change the DNA in the crop species that we grow, because by changing DNA, we hope those plants will have traits that are advantageous to us. And so the way the European regulators reacted was to put on one side of a line genetic modification and the moving of genes between species, and on the other, the myriad other technologies that had a long history of use. And so the wording in the legislation is something like track record of safe use. And so if you're using one of those decades-old technologies to induce a genetic change, then that goes through a different regulatory path than if you're using one of the emerged in the 80s and early 90s genetic modification technology. And that's when you become a GMO, and that's when you have to go through the very onerous regulatory assessment process for a GMO.
Which in practical terms in Europe, has meant, practically that not...
There's one approved... So Europe is a funny place because there's one approved GMO for cultivation, which is a variety of maize, which is grown in tiny amounts in Spain. And then there's a separate regulatory system for importing GMOs. And actually Europe imports lots of GMOs, primarily soya to feed to animals. So it does have this duality that we don't want to grow them here, but we're happy to import them from other places.
Helen, I want to turn to you and really ask you, as a historian that's looked at the use of science in crop breeding during the 20th century, how would you say the kinds of issues that Jonny's been raising, how deep are the roots of those public concerns and kind of regulatory issues in crop breeding? Do we see some of these issues arising, early in the 20th century, or is this really a very new set of concerns?
So I think one of the things I want to call attention to, it's really interesting that Jonny in his comments used the phrase accelerate evolution to describe one of the tracks of work in gene editing as a way of understanding it, because this idea of being able to speed up the the genetic changes within crop varieties is actually an ambition that you see going right back to the start of the 20th century and really connected to especially changes in genetic science and knowledge of heredity and the ways it could be used, especially around the introduction of Mendelian genetics. So the claim about simply understanding a bit better how genes move from generation to generation, how they recombine, and how you might be able to predict and manage that change had already given risen to the idea that we were potentially able to accelerate evolution. And I think we can talk about the different technologies that were introduced in the 20th century in pursuit of that goal. But I think that the really important sort of general story is to think that, that idea of accelerating the evolution of a particular crop variety. And I think that's a kind of crude way of saying, hoping to speed up the rate at which changes appear and can be taken advantage of by breeders. That's really actually a kind of unifying ambition. So when we think about the kinds of concerns that arise around different technologies, I think it's important to recognize that the ambition of intervening in precisely this way has been around a long time. Then with respect to the question about concerns, there is, as I've just said, a different set of tools that emerges at different kind of moments over the course of the 20th century, from tools for inducing mutation, different chemicals or radiation treatments, to the recombinant DNA technologies that Jonny was talking about, to the gene editing that we have today. And different of those tools have provoked different kinds of concerns along the way. I think one way to kind of mark a real shift is to think about what happened, especially in the 1970s and 80s, in terms of activists coalescing around new molecular biological technologies more generally, not just their use in agriculture, as a potentially unwanted kind of scientific intervention, and the ways in which that was connected both to the environmental movement at the time and to the kind of leftist radical movement more generally. And so I think it's important to recognize that some of the concerns people have had have been really tied to broader political concerns and social concerns, and not just concerns about the technology themselves. So maybe that's one way to sort of dive into that question, maybe in more detail.
I mean, that's very interesting, sort of to mark both the sort of continuity in terms of ambitions to speed up or control evolution for the benefit of agriculture, and then some of the disruptions in terms of both what's made possible by the technology, but also how it's received. So, I mean, I want to ask you a little bit perhaps about the 1990s when, as Jonny said, the first commercial crops were using GM, recombinant DNA technologies, were on the market. And that was at a time perhaps when there was a sense that there was a sort of corporate control, concentration of corporate power in agriculture. And so the role of IP, intellectual property rights, the rise of corporations in the 90s that had ambitions to sort of control and own and make money from vast sort of chunks of the sort of value chain in agriculture. And so that became, I think, some of the issues around which the debates about the technology were circulating. Were those new concerns in the 1990s? I mean, what was new about those discussions, do you think, then? Or again, were they really, do you think, more the result of long-running kind of ambitions?
I don't think those concerns that you've just mentioned were new in the 1990s. I do think that the seeds of that discontent with the state of increasingly globalized agriculture and agro-industry, really, you can place that in the 1970s and maybe the late 1960s, these major shifts that happened in the decades after World War II, in which increasingly corporations became involved in restructuring agriculture, restructuring grain markets in really significant ways. And so sort of was pointing to this earlier, the first really rumblings of discontent specific to genetic technologies were actually tied up with critiques of industrial agriculture and what it had become, and with especially the idea that changes in intellectual property governing plants, which had first been put in place in the 1960s in an increasingly international way in Europe, but then by 1970 were becoming also sort of harmonizing with the United States and with other countries, so transnationalizing in a way. Those shifts are really, I think, best understood as happening actually a couple decades earlier. And so the 1990s, when genetically modified crops finally appear on the market, it is with this buildup of discontent, and actually with a quite significant and sophisticated activist network already operating kind of to contest these issues. I think actually one of the things that I would point out with respect to rumblings about or contests over genetic technologies, Jonny was speaking about, I think, the UK and the EU context. In my own research, I've worked much more on the Mexican debates over using, importing, growing genetically modified crops. And there, the explosion of anger and kind of refusal of these technologies also was tied up with the North American Free Trade Agreement and its implementation and what it would mean to have all this GM corn from the United States flood Mexico. And I think this really points to the way in which free trade, neoliberalism, certain kind of corporate control and dominance of the marketplace, and even what states do, is really at the core of the concerns that we've seen.
Yeah, I mean, that isn't the other dynamic, isn't it, about how we saw in the early 2000s different kind of regulatory regimes respond to GM technologies, created a kind of different geography of agriculture and the trade in commodities, with Europe and the United States sort of going in different directions. Obviously, the moment at which the question about genome editing and how it's regulated is kind of coming to the fore is a moment when questions about trade and trade relations are also being discussed and contested. So Giles, taking a step back, I wonder if you could tell us a little bit about genome editing, the science and the technology that's really burgeoned over the last few years, and what kind of opportunities that opens up to you as a plant scientist.
Yes, so what genome editing does is create genetic diversity, and the end result is identical to what one can achieve potentially through traditional breeding approaches that have been underpinning our crop plants production for many decades now. But the one difference with genome editing is that it uses a molecular machinery that comes from bacteria to create that genetic diversity. So once you've got the edited, your genome edited, the machinery from bacteria has a targets changes to the genome, and you can target very precisely to specific sites in the genome. Once you've done the editing, you can breed out all of the bacterial machinery. It's no longer necessary, and you just have a stable event, which is the edited genome in your native crop species. So the end result is very similar to what we would do historically. You would be using genetic diversity that may already exist in the crop plant that comes through mutagenesis, or you induce mutagenesis through gamma ray radiation or chemical mutagenesis to increase the rate of mutagenesis. But the difference between all of those approaches is it's not directed. You have to just randomly mutagenize the genome, and then you try to select for the bits that are useful and improve your crop plants. Now you can do a very targeted alterations to the genome at very precise locations. You don't have this random mutagenesis occurring across the genome. It has two major advantages. You have a lot less changes happening. You have a lot less uncertainty, which you do get when you have the induced mutagenesis. And because it's targeted, we as scientists have a lot more opportunities to really say, okay, this is the precise bit of the gene or promoter that we want to change, and we want to change it in this precise manner.
So when people talk about CRISPR-Cas9, what is CRISPR-Cas9, and is that sort of integral to what you're talking about in terms
Yes,
of...
exactly. So CRISPR-Cas9 is the bacterial machinery. So it's a protein complex that uses bits of RNA that guides it to the genome, the DNA, and then the Cas9 is the enzyme that actually makes the alterations in the DNA, guided by a small piece of RNA. So what you do is you introduce that Cas9 machinery alongside the CRISPR, which is the guide RNA. So you have your guide RNA, which is how you target it to the genome, along with the machinery. This originates from bacteria. You transform that into the plant in a way that is analogous to what you do with genetic modification, where you're introducing foreign DNA in. But the key difference here is you don't need the foreign DNA anymore once it's done its job. And that's a big differential between what we consider a traditionally genetically modified plant, where you have foreign DNA stays in the crop plant that you then grow. Whereas with genome editing, the foreign DNA that you introduce, you can get rid of, and you do get rid of that. And all you're left with is the mark in the genome that you directed as a result of that Cas9 CRISPR introduction.
So do you know, that's the key difference between the sort of GM soya and exactly. maize that we saw introduced commercially in the 1990s and what's potentially possible with genome editing?
Exactly. So the key difference is whether you still have foreign DNA in the crop plant that you grow in the field or whether you do not. And with the genome editing, you no longer need that foreign DNA. And the diversity within the genome, the targeted genome editing that you can create that by other means, as I said, either through exploring the diversity that's already in the crop plant or by inducing diversity through mutagenesis. So the end result is very similar to what we've been doing for decades in the improvement of crop plants, so actually hundreds of years in the improvement of crop plants. But here we can do it in a very targeted manner.
But you could, if there was some advantage to the crop from having DNA from another species after the genome editing, that would also be possible.
What is currently being worked on is using the Cas9 CRISPR machinery, not just to edit the genome, but to drop pieces of DNA into it. And so that's a modified form of genome editing where we would leave some big piece of DNA that wasn't there originally, but you can still get rid of the Cas9 CRISPR cassette, that part of the foreign DNA. But the genome editing that certainly DEFRA is considering right now is where you've just modified or mutated, removed a few base pairs as a result of the action of you haven't introduced any new DNA.
So in terms of your work as a scientist, what kind of horizons or possibilities does this new technology open up for you?
I mean, it's extremely exciting because of it's targeted. We know, for instance, in my research, we know a lot of genes that control how plants engage with beneficial microorganisms like bacteria and fungi that help the plant to acquire nutrients in a sustainable manner. And we can now, historically, in order to try to utilize those genes to breed them into crop plants, we would have had to have done a very large random mutagenesis search for anything that happens to land in that tiny bit of DNA in this huge genome that we're interested in. Now we can do directed mutagenesis at precise sites that we also are quite informed about. So we spent years working on these genes and we know aspects of how they're regulated. So we can subtly change their regulation such that we can improve the sustainability of the system, get nutrients, engage the crop plants to recruit these microorganisms for sustainable nutrition rather than fertilization. So it really is, it opens the horizons with regards to what we can do. And really increases the pace of that activity because it's targeted instead of random. But I also think the end product you end up with, it's a very targeted event. Whereas if you do the random mutagenesis, there's always a lot of unknowns of what else is happening in the genome. You try to breed a lot of that away, but there's always going to be some of those mutations you're unaware of. So I think the products that we can produce with gene editing have a real advantage of being very, very precise. And also from a scientific perspective, they allow us to have a very targeted alterations in the genome in a way we simply could not do in the past.
So, I mean, I wonder if you could be sort of concrete in kind of particular research that you feel, the Crop Science Centre might be kind of considering. technology?
Absolutely. So, I mean, an area that we're looking at right now is plants engage with these beneficial fungi that help in sustainable uptake of nitrogen and phosphorus, the key ingredients of fertilizers. These are critical, these mycorrhizal fungi. They're critical in natural ecosystems. In our arable farming systems where we use a lot of fertilizers, we really don't. The plants do not engage very robustly with these fungi. What we're trying to do is to increase the utilization of those fungi in agriculture to improve the sustainability of nutrition in the agricultural system, so reducing our dependence on chemical fertilizers. And we know in quite detail the genes that are necessary that say when and how the plants engage with those fungi. And so we can do very targeted alterations with genome editing to the promoter elements to optimize those genes for an agricultural context. Because most of the plants are wired over millions of years of evolution of competing in a natural environment. Agriculture is a man-made environment. So we're always trying to optimize our crop plants for this man-made environment. You know, what we have in our man-made environment currently is heavy dependence on chemical inputs. And if we're going to change that and switch that into a more natural and sustainable way of producing our food, then we become much more reliant on what plants can naturally do. And we can change that subtly such that we can optimize things like these natural fungal associations in an agricultural context to reduce our dependence on chemical inputs.
So obviously, one of the reasons why we're talking about this topic now is that in January, the UK's Department for Environment, Food and Rural Affairs launched a consultation about how gene editing should be regulated. The use of gene editing in crops and in the food system should be regulated. And the question is this new technology, which we've seen just mushroom in laboratories and in various applications in the life sciences over recent years, should this be thought of as a development within GMO approaches, therefore regulated as GMOs are regulated, or is this different because it enables different kinds of genetic interventions, therefore should be thought of differently? So, Jonny, just briefly, what are the kind of main contours of this debate at the moment?
Partly the reason we're having the debate is because it's both. And so you can use genome editing both to accelerate the evolution of genetic changes within a species, which is more analogous to those technologies with decades of a track record of usage, or you can use it to move genes between species. And so many other parts of the world have looked at genome editing and said, if you are using it to change the genes within a species, then we're not going to treat that as GMO. And that's the proposal included in that DEFRA consultation. So it's slightly odd that they're opening this consultation with a proposed endpoint.
Yes. Well, this is what we're going to discuss for the rest of this series. But that's really helpful kind of introduction to some of the kind of context for this consultation and some of the issues that it's really bringing to the fore. Thanks, Jonny. I wanted to now sort of think about how the current consultation from DEFRA, what sort of questions that opens up. And what that means for you, Giles, as a practicing plant scientist, thinking about how your science can make a contribution to food security and environmental sustainability. As Jonny said, part of the context for public controversy around GM foods in the late 90s and early 2000s was, at least in the UK, was the fallout from the BSE crisis, the mad cow crisis, and a loss of public confidence in the regulation of the food system. But more than that, there were some other kind of underlying public issues and concerns that were to the fore. And I mean, to boil them down, we could identify three. One being around unintended consequences. You know, if the narrative is this is a powerful new technology that can change the world with, you know, huge potential upsides, then people ask the question, well, okay, but what about the unintended consequences that we assume come along with any new technology? Who's looking out for them? How are they going to be managed? The second set of questions around, you know, who is benefiting? And then a third set of questions around sort of control. Who's calling the shots? So those were clearly three sets of concerns that shaped the public controversy now 20 years ago. Do you see those underlying concerns coming to the fore again with respect to genome editing and the discussions we can expect around genome editing, or do you think it's quite a different set of debates that we can expect to see?
I would hope the debate's different this time around, and it should be. But I guess the challenge is to have the public understand genome editing and recognize that it is a safe technology. And in fact, it's a lot more precise than what we've ever done in the past. So there's a far fewer unintended consequences when you use genome editing, because you can really target the genetic alterations to precisely the areas that you want to target. Whereas in the past, when it was random mutagenesis, which was used extensively in breeding, it is random. You get mutations all over the genome and you have to search for the ones that have relevance, but it's hard to get rid of all of those other mutations. So in my opinion, the technology that we're describing right now is actually far, far safer than what we've had in the past. And the other thing that's changed is the phenomenal advances that have been made in genome sequencing. That means that now we can just sequence the whole genome and tell very precisely exactly what's changed and whether there are any additional mutations that the CRISPR may have brought on board. We can identify and we can breed them out so that we only have the intended mutations that were there. And the end result of the technology, as I said, is really no different to what we've been doing for hundreds of years in traditional plant breeding. So I do think, and certainly the way that DEFRA is presenting it is presented as a breeding technology, and I think that that's correct, because really it's just another modality of breeding, where we're utilizing diversity to improve crop plants. I think that point of who controls the technology is probably the most important one. In fact, the reaction to genetic modification has created an extremely onerous process to get approval of crops that is extraordinarily expensive. And what that has actually meant is that only the largest corporations can use genetic modification, because they're the only ones with deep enough pockets to create products on the market that go through all of those approval processes. I think this is rather an irony, because I don't think it's what was intended, but the degree of regulation is such that we've actually put the technologies into the hands of very few corporations that can afford to use it. And I think that was actually an unintended consequence of the approach that was taken. With genome editing, because it's not genetic modification with regard to the fact that it does not leave foreign DNA in the plant, I do not believe it should be regulated in the process by which traditional genetic modification is regulated. And if that is the outcome of this consultation, then the technology becomes much more freely available to a lot more individuals, not just large biotech companies, but smaller companies as well. But also, it makes it much more open to us in the academic world to realistically be taking forwards products that can go into breeding programs. So it really takes away the total control of the technology from a few very powerful corporations to many more individuals.
Of course, with any intervention, there are always, potential unintended consequences. So, for example, you said that the power of sequencing would enable anyone using genome editing to go back and check and see if there had been any changes that hadn't been intended. Do you think that would require an additional kind of regulatory step to ensure that happens?
I think it would. I mean, if you're looking to make a product and you want the public to have confidence in that product, I think you could put a regulatory step in there. It's not an onerous step to sequence the genome of the plant that you finally have, and to demonstrate that the only mutations that are induced are the ones that were at the target site. So that's intended consequences as opposed to this unintended consequences. And the challenge of unintended consequences, if you need a point of proof, and I think in this regard, it's a sequence of the genome is your point of proof that you have only changed change the site that you intended to change.
I can anticipate another set of sort of questions or concerns, which really stems from the very kind of power and utility of this technology, which is the sense that if, it's been demonstrated in the lab to be amazingly versatile, do we, the public, need to wonder, to what ends this technology is going to be put, and think about how that is controlled or regulated from the public interest point of view. If it becomes sort of much more widely used, much more readily used in labs around the world, do we need to think about, what the intent in the minds of the technologists is, and how we sort of think about controlling?
I think what you want to regulate is the changes, the intended consequences, to ensure that what you're changing is safe, as opposed to, because, as I said, gene editing creates the same product as more traditional breeding approaches do. But for traditional breeding approaches, there's actually fewer checks and balances than there are if you certainly use genetic modification right now, and we see with regard to gene editing. But to have a whole load of regulation because of how you've got to that end result makes little sense. You need to regulate and consider the end result as opposed to how you got there. Because if you get to the end result by gene editing, and you can achieve the same end result by chemical mutagenesis, exactly the same end result, why would you regulate a product that's been made by gene editing differently to how you'd regulate a product that's been created by a more traditional approach? Because the end result is the same. So it's the end result you want to consider. What is the consequences of what you changed? And is that safe? And is it good? And is it beneficial?
Do you think we have an appropriate kind of governance and regulatory system to think about the end result at the moment?
Well, yes. So there's certainly there's processes that look at the behavior of new varieties of crops and their safety. For instance, is there new allergens being introduced? Is food safety issues and the performance of that particular variety in the field. And we have a very strong regulatory process here in the UK with regard to how we introduce new varieties and approve them for growth by farmers. And I don't see that a gene edited product should be treated any differently than the processes that we already have in place.
Great. I mean, if obviously we will see, over the next few months how that consultation goes and what kind of reactions, are generated. If it turns out that there is a strong public feeling that genome editing should be considered in the same way that genetic GM technologies are currently considered, where do you think that leaves the kind of research you're doing and the kind of possibilities that you see for plant sciences? Yeah,
that would be extremely limiting. I mean, I think already it's frustrating to have the incredible restrictions on genetic modification that essentially has stopped that technology from certainly being used in Europe, and is really limiting its utilization globally, or certainly making it much harder. If we end up with the same result for gene editing, then the possibility to innovate in crops is really extremely limited. We're holding back the improvement of crops to the technologies that were available to us 40, 50 years ago. You don't have this in medical sciences, but in plant sciences, what we can actually produce as products is still limited to what we could do 40, 50 years ago. But when you look forwards, we're going to have a population of 10 billion people with a lot more meat consumption. If we just take a business as usual scenario right now, we're going to use a lot more chemical inputs, a lot more insecticides, a lot more fertilizers. We're going to cut down more of the Amazon rainforest. We're going to see greater impact of agriculture on the environment than it already is. And agriculture has a massive impact on the environment already. Significant contributor to greenhouse gas emissions, a significant contributor to land use changes that impact biodiversity, and a significant contributor to the release of nitrogen and phosphorus into our aquatic and terrestrial ecosystem. So we want to do that differently, right? We want to, come 2050, have a system by which we grow our food that does not have those huge environmental impacts, and can meet the needs of a large and quite demanding global population. And if we just take a business as usual scenario, I think we're basically sacrificing the Amazon rainforest to the mass production of agricultural commodities. Now, we can't, some compromise has to happen here. You either allow us to innovate, to create opportunities to change that system, or you simply have to accept that it's a business as usual scenario. And a business as usual scenario is going to be more chemicals and more land use changes into agriculture.
Yeah, this is going to be fascinating to watch over the next few months. It seems to me a possible outcome will be that, to some extent, if it appears that the choice that's sort of being offered the public is either these new technologies should be thought of in the same way that conventional breeding technologies are thought of, or as GM technologies are currently thought of, we shall see. But I mean, I'm not convinced that the answer will come back other than, these should be considered in the same way that GM technologies have been considered, if that's how the choice is presented. If the choice is opened up to being innovation is going to be needed in agriculture and it needs to address these sorts of big challenges and big opportunities, how should what should a new regulatory and governance system look like that achieves that end? If that's the terms of the debate, I think we may get a different answer. Mm-hmm. So that may be a challenge that we're left with. I'm not quite convinced that the debate is currently being set up in a way that's going to encourage that richer discussion. But time will tell. I
mean, I'm excited by the potential I think we have to rethink the way we approach our food production. It's not sustainable. It does cause issues within the environment. But I'm actually very excited about the opportunities, the new interests in conservation agriculture. And I think there are areas where we in the scientific world can contribute to a much more sustainable and more equitable global food production systems. We can only do so if we have the capability to innovate. And if our hands are continuously tied, then it's very hard to contribute to that as scientists.
So thank you all for participating in this episode, and thank you for listening. Join us next week when we're going to be looking more closely at the particular applications of genome editing and genetic technologies in agriculture. CSaP's Science and Policy podcast is a production of the Centre for Science and Policy at the University of Cambridge. The series is hosted by me, Dr. Rob Doubleday, and is produced by Kate McNeil, with the support of research assistant Alice Millington. You can learn more about CSaP's work by visiting us on Twitter at CSciPol, or by visiting our website at www.csap.cam.ac.uk. If you've any feedback about this episode, or questions you'd like to address in the future, please email us at inquiries@csap.cam.ac.uk. Thanks for listening.